let's go up to here for a
I
moment.
gotcha. So if I have an intermittence loads file that has 25 entries in it and they're all in one file, I still need the rest of the files named.
That's
even
correct.
though they'll never be opened.
That's correct.
So,
Yes.
so in this case, actually the hydro conditions, the load, the boundary is repeating every year.
Yeah.
Saying, okay, if I use different one, I just give it different name.
Yeah. Yeah, as you've noted, so the code knows which files are constant and which ones are time varying, or which ones are read once and which are time varying. So up here, if you say I have 11, every place that this is a time variable file, you need 11 of them. Even if they never, well, okay, you'll need 11 of them. And as is noted, in this particular case, we're going to repeat the hydrodynamics, the same hydrodynamics 10 times. So what happens is you need to specify that file 11, because what it's going to do is it's going to close this file and open the next file. Now, in this case, it's going to close it and open the same file again, but it's going to do that. Even if you have, if you have one hydro file with annual flows or whatever, you'll need to, well, there you go. You'll need to specify it, and it'll close it and open it, close it and open it, close it and open it.
So that's why the files always start with 0, right?
Yeah.
Yeah.
Well, in this particular case, because you're running the same one, but if you had different files, I would have done it a little differently, but it is the way it works.
Yeah.
Yeah, okay.
And then you get, after you've finished with your inputs, then you have your output files. The output files are the only one, singular, So, so if we ran 10 years, you don't get 10 output files, you get 1. There are a couple of key files. This one is SNP, that's called the snapshot file. That puts out in ASCII all the model results for all state variables at intervals which you specify. You could imagine that for a Bay program model with 59,000 cells and 28 state variables, that file gets monstrous. So you probably don't want to put it out too often. Again, I think this harks back to days when, grids were smaller and you could actually read the for the 30 box model, you can read and interpret the snapshot file. And in fact, for simplicity, when we run the 30 box model, we're going to work with the snapshot file for simplicity. There are also 2 more key files, the plot and the APL. These are binary files. And these are really the key output files. The plot file contains, once again, all state variables at all locations, output at arbitrary intervals. And if you're going to do graphics or anything like that, you probably, you work with the plot file. And the outputs are, the intervals are arbitrary. The APL is exactly the same. The difference is the values in PLT are instantaneous. If you tell it I want the outputs every day, you'll get the exact outputs at 0, 1.0, 2.0, 3.0. What APL does is it averages the model results over arbitrary intervals. So if you say, for example, I want the APL every day, you'll get the average of day 1, the average of day 2, the average of day 3. You can also do things like take 30-day averages. I want the monthly average. You could average them over seasons. You could get a summer average. So, but these are the 2 big ones. And what you need, of course, now is you need a program to read either PLT or APL, and then you need a plot file, something to plot them. We're really not going to do that in this class. That's kind of up to you. I mean, depending on what program you like to use to do plots and how you like to plot them and that sort of thing. So we're not going to do that.
Quick question?
Yeah, go ahead.
Sorry to interrupt. The snaps, are they averaged or instantaneous?
Instantaneous.
Instantaneous, okay.
Yeah. I'll mention, depending on, maybe some people find making those plot files really easy, but remember that what comes out is the cell number and the concentration. Cell number. You don't know where those cells are, right? Because this model contains no information. So when you start wanting to do, nice, I don't know what, nice visuals of the surface layer of Chesapeake Bay or the bottom of Chesapeake Bay or something like that, you need an external file that's going to tell the plot program where those cells are. And it may read the CH3D grid, typically we get the coordinates of the cells from the hydrodynamic team and we enter those separately. But, these are things for you folks to work out. And once again, you all have different graphic programs that you love or favor or something like that. So you have to work, you're going to have to develop that interface. I'm sure that whatever we do with the Bay model, I'll make available to you, or Richard can make available to you, and you can go from there if you're specifically interested in Chesapeake Bay. Anything else I need to go over? We're running into lunch in a couple of minutes, so...
And, I'm sorry, one more quick question.
Yeah, go ahead.
The PLT and the APL or whatever it was, are they just a water column, or is there another file for sediment output?
They are all active variables, so the sediment outputs are in there.
All right, so in that numbering scheme, your map file, do you include what the sediment segments are? How does that...
The sediment segments have the same numbers as the surface boxes.
As the surface box, okay.
As the surface boxes.
But it would show up, it would be like the number of water cells and then the group of sediment cells after that. Okay.
Yeah.
No. You have to decipher the output. It's a mess. I wrote a program to do that. You're welcome to it.
No, but I mean, the binary file, he's got all the water column cells, and then he's got the sediment cells sort of following it in sequence. Correct. Right. Well, he's shaking his head yes, but...
What was the problem, Dan?
At least the APL file has, for the sediments,
I'm talking about the sediment bed, the nutrient fluxes in the bed or
right.
stuff. Okay.
Those are written out as independent variables. There's a common block that's basically, all those are written out as blocks. Okay. So it's one variable at a time. It's not written out by cell, it's written out by variable. Okay. And so you have to decode, that's what's, I just did it, so that's what's in the APL file. Okay. All right? Yeah. And decoding it is a pain in the butt. And there is a map translation file that Richard has that we use to get it for the current version of the Chesapeake Bay model. Okay. And it's keeping track of the indices is a challenge. Yes. Very difficult.
Here's the... here's actually the but funny because when I opened the main, the first line it went to, now I'm looking at the right-hand screen, guys. The first line it went to was right plot. So I must have been looking at this fairly recently, and there you see that what it does is it writes out the sediment model state variables from 1 to the number of surface boxes. Now, the problem is, in a sense, if you just have a plot file, all you know is there are arrays written out that have NSB contents. You don't know that the first one is DOC, the second one is ammonium, the third one is nitrate. Right. You have to have this code basically to say, I want to look at sediment nitrate. But if you want to know where they are, they're written from 1 to NSB. And the first nitrate is under model surface cell 1, the second nitrate. Right, okay. And actually it's similar, if we go up to this one, up here, this is where it writes the model the actual concentrations in the water column, and you see it writes concentration, the number of active constituents. So again, what you're really going to get there are 28 arrays, and it's up to you to know that number 27 is oxygen, something like that. You don't know.
So another quick question. You don't do variable stoichiometry anymore, is that correct?
That's correct.
Okay, so that's, all right.
But we'll talk a little bit, but you still need to know carbon to chlorophyll ratio,
Yes, right,
because algae are carbon, but your data is chlorophyll, so that's why it writes out the carbon to chlorophyll. You know, it tends to write out, I won't go into this in great detail, but at the heads of the plot in the APL file, it will write out information that you will need to do the plots. Like it'll probably write out the number of boxes and that sort of thing. And here it's writing out some information you need to interpret the to interpret the algal biomass. But you know, clearly, yeah, you need this bit of code here to see what's being written out. Otherwise, there's no identity. You know, there's no identity in those APL and PLT files. They're just variables, and it's up to you to know what the order is and what you're plotting. We've probably, I don't know if there's much more to say, really. You know, the control file is extensive. We're going to look at some other of the contents when we actually get down to running the model. Like I said, I don't want to go over every one of these. It's in the manual. And but I think we've covered, you know, the things that are important. I think we've covered what you need to know. So before we knock off, the last opportunity to, last chance for questions. Actually, you know what? If you come to me later today with a question, I'll probably help you out. But you know, yeah, serious. Okay, so we're going to break for lunch.
One quick question. So for the APL, like you said, you can do arbitrary interval. So there's a place for you?
Yeah, it's in the, actually, it's open to it right here, average plot frequency. So, and in fact, you can even vary them. Tom has the facility to vary them.
Okay. So you can do like 20 days, then from day 1 to day something, then...
That's correct. You can do exactly that. And it's done in this, average plots are on, so we're going to put out that file. We're going to start, we're going to specify the interval from day 0, and it's going to be that. Here you say we only have one interval. These two right now are dummies. If I had put 2, then it you know, would have changed to 10-day intervals at some point, but that's how it's done.
Oh, I didn't realize that. So you can actually get out more than one interval for average plots and produce several APLs.
Yeah.
Ah,
No, it only produces one APL, but the interval at which outputs are averaged, you can change.
So like you define how many
but wait,
like, time periods you want
so
to have.
the number of APL files there is 1. Suppose that was 2. Wouldn't it write 2 files?
No, this is how often you change the interval, or how often you specify the interval. In this case, we specify it once.
Gotcha.